Hello everyone, my name is Guo, and I'm VP of Data Science and Bioinformatics at Crown Bioscience, a preclinical contract research organization providing high-quality models, technologies, and support for your drug development needs. Today's 15-minute webinar is part of our educational tech talk series focused on in vivo solutions for oncology and immuno-oncology drug development. There will be no Q&A session at the end, but please feel free to ask questions in the chat box, and we will answer them as quickly as possible. In my talk titled Predicting Clinical Response with Mouse Clinical Trials, you will learn mouse clinical trial design and methodology, drug efficacy evaluation, biomarker discovery, and MOA investigation. There are many in vivo models for oncology drug discovery, including patient-derived xenograft or PDX, cell line-derived xenograft or CDX, syngeneic models, and genetic engineered mouse models, or can be used in mouse clinical trials. So what is a mouse clinical trial or MCT? Well, MCT is a phase 2-like in vivo study that is aimed to evaluate drug efficacy. Let's use PDX as an example. In this PDX mouse clinical trial, a patient can conceptually join both arms by way of PDX. Here's how. In this illustration, there are 6 PDX as represented by the 6 colors. For each PDX, we allocate 3 mice to the first arm, 3 to the second arm. This is a so-called 3-to-3 design. So you say the 6 PDX capture inter-tumor heterogeneity, and the 3 mice of PDX measure inter-tumor heterogeneity within both arms. Furthermore, since we have mice of the same PDX in both arms, they themselves can serve as control across arms for better measurement of efficacy that can also help biomarker discovery. In practice, we don't need to start the inoculation at the same time, but rather randomly enroll a mouse to an arm when its tumor volume reaches a certain size, say between 100 and 300 cubic millimeters. So it is a randomized control trial. Once the trial starts, tumor volume are measured twice every week, or even more frequently for a few weeks or a few months. Since we have genomically profiled the mouse models, MCT can also be used to discover predictive biomarkers and investigate drug mechanism. Let's take a look at tumor growth curves from some mouse clinical trials. This is a 42 PDX mouse clinical trial. Left shows tumor growth curves in the vehicle or control group, and right is in the cisplatin treatment group. Horizontal axis shows days after inoculation, and the vertical axis is a log-transformed tumor volume. Each curve is a mouse, and we do observe variations within and between PDX in both groups. This is a larger 90 PDX mouse clinical trial, again, with variations in growth curves, especially in the drug treatment cetuximab group. With such tumor growth data and the model genomics data, what can we do? Live approaches can be used, such as calculating correlation between drug efficacy and the gene expression. But more rational and systematic approaches give much more insights, as we detailed in this paper, with some illustrations in subsequent slides. Briefly, we will touch upon the 4 topics: drug efficacy, study design, biomarker discovery, and MOA. Let's start with a very basic drug efficacy evaluation. Traditionally, drug efficacy is measured by tumor growth inhibition, or TGI. Simply put, we stick with a specific day, usually the last day of the study, and calculate tumor volume changes from the starting day for both treatment group and the control group. and then basically the delta T and the delta C here, and then use this formula to get DGI. Of course, there are some other variants of DGI. The problem with DGI is that it is both tumor growth rate and time dependent. It always gets close to 1 by day, as shown in the two graphs here. So in summary, TGI is not a stable efficacy endpoint and is biased. To remedy this problem, we proposed an unbiased efficacy endpoint called AUC ratio. Not reading the details, basically this endpoint is a ratio of tumor growth rate between drug treatment group and vehicle control group. Hence, as time goes by, it actually approaches to a stable constant, as shown in the two graphs here. There is yet more powerful approaches that go beyond a single number efficacy endpoint. One such approach is to model a muscular trial as a clustered longitudinal study by linear mixed model. The good thing about linear mixed model is that it uses tumor volume data for all days from all mice. Therefore, maximize information and data utilization and more accurately measures drug efficacy and performs biomarker discoveries, as we show in the next slides. First, on biomarker discovery with an emphasis on study design. It has been a frequently asked question for how to determine the number of PDX and the number of mice per PDX in a muscular trial. From a statistical point of view, as long as the total number of mice is the same, different designs have comparable statistical power. So, for example, if there are 150 mice in an arm, we can do 30 PDX each with 5 mice, or 50 PDX each with 3 mice, or even 150 PDX each with just 1 mouse. With a good number of mice, there is not that much concern on statistical power, but rather the purpose of an MCT dictates the study design. Specifically, for biomarker discovery MCT, it is advisable to have more PDX and fewer mice per PDX to the extreme case of just using 1 mouse. This is for better genomic and population representation, which is vitally important actually for biomarker discovery. For biomarker validation, it is nice to have fewer but selected PDX and more mice per PDX for more accurate measurement of efficacy. I will now illustrate MCT-based biomarker discovery using my favorite example, cetuximab treatment on gastric PDX models. Cetuximab is an EGFR monoclonal antibody, so PDX with higher EGFR expression have better response. Lowering the drug target, when we use DGI as the efficacy endpoint, we observe a good correlation between DGI and EGFR mRNA expression. But there are 156 genes with better correlation. When using AUC ratio, we say that EGFR goes up to the second place with a much stronger correlation. Finally, when we step up to linear mixed model, EGFR becomes the unchallenged top pick. So, more sophisticated methods do lead to a clearly favored candidate in biomarker selection. Here's another example. Unlike cetuximab, in this example, we knew nothing about the drug target and the mechanism when performing the study. So it is a so-called hypothesis-free biomarker discovery exercise. We classified the 77 PDX into responders and non-responders based on AUC ratio, then discovered 6 PDX, 6 biomarkers. The first 4 are mutation biomarkers, and the last 2 are gene expression-based biomarkers. For example, for the first 3 biomarkers, the mutants are resistant to drug treatment. Also, the 2 expression-based biomarkers turned out to actually measure the activity of the pathway targeted by the drug. So everything makes sense. Two of the mutation biomarkers were later used in clinical trials for selecting proper patients. Okay, moving to MOA investigation. We first use a 16 PDX mouse clinical trial on irinotecan, which is a DNA topoisomerase 1 inhibitor that interrupts cell cycle in the S phase by irreversibly arresting the replication fork, hence stops DNA replication initiation and cause cell death. We identified the genes whose expressions are correlated with drug response by three methods: TGI, RECIST, and the linear mixed models. It turns out that the genes identified by linear mixed models are highly enriched in DNA replication initiation and also in cell cycle based on geo and pathway enrichment analysis. So precisely the mechanism of the drug, but not so for the other two naive approaches. A highly enriched protein-protein interaction network consists of 23 genes in the top 100 genes selected by linear mixed model is also shown to be highly enriched for cell cycle. Here is another example. We tested efficacy of anti-PD-1, anti-PD-L1, and anti-CTLA-4 in 12 syngeneic models in our meal screen studies. They responded differently. H22 is the most responsive one, and B16-B06, which is the melanoma model, is the most resistant syngeneic model. We have proteomics data for all 12 syngeneic models. So we performed a factor analysis and identified several factors associated with drug response. For example, proteins with high factor 1 scores are upregulated for oxidative response and downregulated for interferon pathways. On the other hand, for proteins with high factor 2 scores, they're upregulated for cell cycle and allograft rejection, and downregulated for EMT. So you say mouse clinical trial is really a powerful tool for evaluating drug efficacy, discovering biomarkers, and investigating the mechanism of drug. It can be conducted on many in vivo mouse models, including PDX, CDX, syngeneic, mu-prime, or even a mix of different types, such as PDX and CDX. We have done that before. Mouse clinical trials can be designed based on study purpose. Certainly, they can be used for evaluating drug efficacy, but for biomarkers, we can have specialized design for whether it is biomarker discovery or biomarker validation. And there are different efficacy endpoints. We proposed this AUC ratio as an unbiased endpoint and demonstrate that it's actually much better than the traditional use of TGI. But further, statistical modeling approaches, such as linear mixed models, can even more effectively evaluate drug efficacy, elucidate drug mechanism, and discover predictive biomarkers. Thank you everyone for participating in today's Tech Talk. I hope you have enjoyed it, and join us for the next talk in the series, titled Adding Value to Drug Development Through In Vivo Model Imaging by my colleague, Dr. Yingfei Yin. To learn more about relevant resources, visit the resources section of the video platform and visit us at crownbell.com. Thank you.